1987. And this is the famous lecture in which he first proposed that information transfer in biology happens from DNA to protein. And at the time, he said never the other way around. Right? And that's pretty much what we know today as well, that it happens from DNA to protein. Soon after this, though, within the next decade or so, it became clear that not only does one sequence of DNA not encode uniquely one set of amino acids, but also that this whole decoding process is very error-prone. Right, so this is the codon, what is sometimes called the universal codon table, not entirely universal, but close. And what we know today is that, for example, although CUU is noted as leucine, CUU can sometimes be read as proline, right? Or UUU phenylalanine can occasionally be read as leucine. Also as leucine. And these rates have been quantified across bacteria, or now Drosophila recently, mammalian, and human systems, also yeast. And all of them have a rate of about 1 in 10,000 amino acids being misread. So 1 in 10 power 4. In bacteria, we also know that in actively growing cells at any given time, about 10% of the total proteome is mistranslated. So it carries proteins that are different in sequence than the underlying genetic sequence. So a few years ago, we asked whether this sequence diversity in proteins means anything for phenotypic variation. Is it also translating to variation in phenotype? Because of course it doesn't have to. And to do this, we used a single cell system, a microfluidics based device, which is sometimes called the mother machine in bacterial work, but it's basically a PDMS silicon-based device where, I don't know if it's visible, but there are these very narrow channels. So essentially, you trap this whole, you start by seeding about 10,000 to 100,000 cells, and you start with this cell, which is called the mother cell at the bottom, and it keeps dividing and dividing, and as the cells enter this, which is the media lane, it will get washed up. So the advantage of the system is that you can track single cell phenotypes, like the length of the cell and how long it takes for doubling and so on. So I'm just showing you one result that we had from there, which I hope will make the point I want to make. The black curve here is wild type. All the colored ones are the wild type, but treated with an amino acid analog, or in this case an antibiotic, to enhance mistranslation rates. I hope you can see that upon enhancing mistranslation rates, the distributions for doubling time all increase. Okay, so we quantified it, which I'm not showing here, and we were able to show that variability in fitness-linked rates, such as doubling time and also cell length, increase significantly when you have mistranslation. And this can be environmentally induced, as I'm showing here, but it can also be genetically induced, which we checked as well. Okay, so just to remind you at this stage, what I mean by mistranslation is decoding that results in an alternate protein sequence, decoding of the DNA. So mistranslation, as you can imagine, just like mutations, is most often deleterious or costly. And this is thought to be mediated by proteins that have the wrong sequence ending up holding the wrong misfolding, also by aggregation. We know from other work that mistranslation can also reduce the benefit of beneficial mutations, the effect size of beneficial mutations. We know from work looking at genomic sequences that there seems to be selection against sequences that promote mistranslation, all of which goes to suggest that mistranslation is probably costly. Contrary to this, though, there is also now substantial evidence in the last decade or so, including our own work, that mistranslation can increase survival and under several conditions. And usually this is mediated by an increase in effective population size, sometimes due to activation of stress responses. So not necessarily a specific response. Just to give you an example of how within generation effects of mistranslation can become important transgenerationally, consider two populations, one which has low mistranslation and another that has higher mistranslation. That's, you can see that these different colors in the amino acids, yellow, red, and so on. And then you have, let's say, some alternate environment, and let's consider it a stressful environment coming up. And people have shown that in such a situation, you can have as drastic an effect as only the higher mistranslation cells surviving and only the others being harmed. This, again, is known across, for example, high oxidative stress environments, specific antibiotics, and so on. So what I've said so far is that mistranslation is typically costly, but also beneficial in specific environments. Which are these environments? Because these are bacteria, one very popular choice has been antibiotics across labs. So what we try to do here is to come up with a broader framework to understand when is it beneficial. So because people have looked at lots of satellite environments, and it's hard to come up with a common framework with that. And we also try to do this, try to understand how does the cost of the external environment that you're imposing feed into this benefit, right? Because mistranslation extracts the cost, but so do antibiotics. So we're looking at this in different ways. I'm going to present one of these today, which is to see how does the cost of mistranslation play out, or does it interact, and if it does, how interact with the cost of an externally added antibiotic. And ultimately, is there a benefit? And if when there is a benefit, how do we make sense of these two costs? So how do they play out? So in order to address this question, we used an experimental evolution regime. And before that, we had to create strains that are mistranslating. So we did this in the following ways that I'll talk about now. You can manipulate mistranslation in E. coli by manipulating components of the protein synthesis machinery. So these are different tRNA genes, and when you reduce the number of a specific kind of tRNA gene, in our case, the initiator tRNA, you can turns out that you get corresponding mistranslation. So we saw more non-AUG initiation. You can make mutations in the ribosome, targeted point mutations, which will increase rates of mistranslation. We also made point mutations in the ribosome that decrease rates, so the other way around, make the ribosomes hyperaccurate. And then using these strains, we looked at growth rate, which is a common measure of fitness in rapidly dividing bacteria in exponential phase. Black is the wild type, and I hope you can see that all of the others are different from the wild type. Two of them are significantly lower. One has huge variability. This one, which is not very different, is the hyperaccurate. That's the one whose accuracy is higher than the wild type. So there is some cost of mistranslation we can quantify if we look at growth rates through relative growth rates. Now in this system, if one also adds antibiotic, again the growth rate, this is the growth rate that is shown here, this one on top at 1.5. Now we're adding 3 different antibiotics, ciprofloxacin, kanamycin, ampicillin. And this line shows how the growth rate drops in each environment. Similarly, I picked the next one, which is red. That's one of the mistranslating strains. Same thing, it has a basal growth rate, which is already lower than wild type because there is a cost of mistranslation. But on top of that, now there is a cost of antibiotic. Right? So similarly, in all the other cases, we therefore quantify, and I'll come to how we quantify an ancestral cost of mistranslation. There's also an ancestral cost of antibiotic exposure. And now, the way we quantify these costs is just to keep it simple. We used growth rates and said that growth rate in antibiotic relative to how it grows in LB, that is when you don't have antibiotic, is a measure of the cost of exposure, and a cost of altered accuracy. And I'm using that word because it's not only mistranslating, it's also hyperaccurate. So altered accuracy is simply relative to the wild type growth rate in LB. These are the measures we use. In both cases, if you have a no cost situation, then the number is 1. So basically, no difference if you grow an antibiotic or LB, or no difference between wild type growth rate and test rate. Okay, so this is the experimental, evolution regime we use. We use 48 well plates. We subculture every 10 generations for 300 generations. So that is when the cells are still in sort of late log phase. And we use 3 different antibiotics that have various modes of action. Ampicillin, this targets the cell wall, ciprofloxacin, DNA repair, and kanamycin, which is involved with protein synthesis. And at the end of this period, we look for signs of adaptation, not only through growth rate, but today I'm only presenting growth rate. We also look at survival and how the minimal inhibitory concentration changes. Okay, and of course we have control lines that are grown in LB without any. So at the end of this process, this is just to show you, this is one example in ciprofloxacin. Each of these strains starts somewhere here on the y-axis, and I hope you can see that everybody increases, and all of these are significant increases. Similar patterns are seen for all the regimes, kanamycin, ampicillin, LB. In some cases, the wild type, which is black, does not show a significant increase, but all of the strains with altered accuracy too. Okay, so now this is growth rate, but what we want to do is look at how the costs are changing. So I'm just showing you 2 strains to not make it too confusing. So this is one of the strains, MIS1. It's a mistranslating strain. What is plotted here is cost of antibiotic on the y-axis and cost of accuracy on the x-axis. So let's look at kanamycin. this MIS1 strain grown in kanamycin has some cost of antibiotic, which is 4.5 or something like that. And this is ancestral. And after evolution, the direction of the arrow shows adapted lines after evolution. I hope you can see that it reaches 1, which is the no cost situation. Similarly, with ciprofloxacin, starts somewhere here, reaches almost 1, ampicillin reaches almost 1. So the antibiotic costs seem to be compensated quite comprehensively and quite fast during adaptation. The accuracy costs, so movement along the x-axis, are also compensated, as I hope you can see. They're all moving to the left, but less. I mean, the effect sizes we see are less than in the case of the antibiotic. Now, interestingly, if you look at the hyperaccurate strain, so this is a mistranslating strain. If you look at a hyperaccurate strain, although it compensates antibiotic costs, that is movement along the y-axis, the cost of accuracy increases. It either doesn't significantly change or it increases. So this suggests that there is some constraint to increasing the levels of mistranslation during adaptation. Right? So we see effects in the opposite direction. So this mistranslating strain acquires mutations that actually make it hyperaccurate in some compensatory way, it seems. But it seems the cell has a constraint to increasing. So it's probably sitting in some value to find it out of it as far as the accuracy rate here is concerned. So we find overall that the effect of genotype dominates over the effect of the environment. There is also a strong interaction effect in these experiments. But before I go to what we see in terms of the mutational basis of this adaptation, there is some prior work that suggests that mistranslation may affect mutation frequency. Right? So one could ask, how do you know the effects you're seeing is because of mistranslation? It may be indirectly through mutation rate being affected. So to test this, we carried out a mutation accumulation experiment. So this is these are E. coli strains, 50 lines of each of these E. coli strains, these 3. And the idea is to essentially evolve them under conditions of drift for a period of time. This is done for about 2 months on a 24-hour basis. And of course, we're not eliminating selection, but it's as minimal as we can make it. Every day we pick a colony from a pre-marked area, transfer it to another plate, so on, so on, so on, so on, and then we sequence all of these 50 lines at the end of this. On average, they have anywhere between 1 to 10 mutations. Right? So we sequence these and identify them, and then we quantify the different kinds of mutations, how many transitions, transversions, how many coding, non-coding, etc., and also the rate, because we know how many lines we have and how many what the doubling time is. And to cut a long story short, for lack of time, the answer is that there is no change. So it turns out that altering translation accuracy does not change the mutation rate of spectrum, which means that we can fairly confidently say whatever effects you're seeing are because of the changes we've made towards increasing or decreasing mistranslation. So coming to the genetic basis of adaptation, these are the 3 antibiotic regimes, CYP, CAN, and AM. So if this is the wild type, the black circles represent what mutations are seen in the wild type. The x-axis has a list of genes. Don't worry about what they are. I just want to highlight the pattern here. So these are the mutations in the wild type in CYP, these in CAN, these in AM. If you compare that now with the high mistranslation lines, we find that there are some overlapping mutations, but also many novel ones that never show up in the wild type. There's a whole bunch of novel mutations that come up. Interestingly, the same thing happens with the hyperaccurate lines, that there are novel mutations as well as overlaps, but they're distinct. Okay, so there are distinct sets of mutations that come up, or distinct mutation trajectories, when you elevate mistranslation versus when you decrease mistranslation. And another strong pattern we see is that there is an increase in mutational diversity. That's not very clear from this figure, but we see much more mutational diversity in the strains with altered accuracy. And also we see large effect changes. So there's a huge increase in antibiotic resistance, even though we've only cultured them in low sub-MIC antibiotic. Right, so this is just, before I end, I just want to show this because I think it's of general, at least it's exciting to me, it's of general interest. These are our control lines. So these are lines that have never seen antibiotic during adaptation, right? But at the end of it, we tested them to see how do they do to now expose them to antibiotic. And very interestingly, in kanamycin and also in ampicillin, we see substantial reduction in antibiotic costs. What does this mean? In other words, that means that these strains that have never seen antibiotic are now doing much better in antibiotics than they did initially. Their MICs are also a lot higher. So the implication for this would be that if you had an infection potentially and you don't treat it with antibiotics for a very long time, then you can be accumulating all sorts of non-target, so we don't see target mutations, non-target mutations, which can nonetheless, I suppose, through all sorts of epistatic interactions, lead to an increase in resistance. So it's a scary thing as well, because they've never seen antibiotic, but you still have strong selection for resistance. Okay, so to conclude, what we find here is that the cost of the environment, in this case antibiotic, dominates over those of the altered accuracy, and they're also rapidly compensated. The altered translation accuracy, whichever direction you go, also alters mutational trajectories seems to be in a fairly reliable way, so we get the same across lines. The mechanism of mistranslation seems to be important. So it's not that no matter how you mistranslate, you end up with the same set of mutations fixed, but it depends how you mistranslate. I think this is a combination of course, some level of epistasis with the background mutation that induces mistranslation, but also a lot of pleiotropic effects, which at this point we're not able to disentangle. Okay, so do I have a minute? Okay, just very quickly, we don't know why the mutations are different in these strains, right? And the only prior sort of framework we have to go on is this work from Andreas Wagner's lab, where they through modeling and also through using their antibiotic resistance gene reporter on a plasmid, they suggest that whatever effects we see of mistranslation on the mutational landscape is probably driven through cost. And the cost they are talking about is the cost that pulls down. So basically a cost because of protein misfolding and aggregation. Right? So at least this work that we currently have seems to show that that is not true, because whether you increase or decrease mistranslation, they're still altering the mutational landscape. Right? So it cannot be driven by the cost of added load from mistranslated proteins. So we don't know what's going on, and this is an open question. We're trying to figure it out. Right, so coming back to this really nice image, of course we have zero evidence that an altered amino acid sequence in any way can direct DNA change. I'm not suggesting that, but I think it's really cool that we have altered proteomes which can maybe lead to predictably altered genotypes. And I'm really excited to see where this goes forward. Okay, so I'll end there. Thank all the people who have been involved in doing the work, conceptualizing it. This is our current 3-year-old lab. These are our funding agencies. And yeah, thank you for your attention.
I do have a question about what is the identity of the mutations that change, because like previous work on mistranslation was showing how, for example, if the mistranslation is adaptive, you would tend to kind of increase the propensity, like the levels of expression, so that you get more of the adaptive product.
Sorry, you have to repeat what was the first part of it?
What are the mutations?
Oh,
are they more like global effects?
Oh, what kinds is it? Okay, so in the antibiotic regimes, they are largely target driven. So if it's ciprofloxacin, it's DNA guidase, which ciprofloxacin binds to. So in the antibiotic regimes, it's largely driven by resistance conferring mutations. In the regimes where I showed without in the LB regime, where there is no exposure, nothing to do with antibiotics, and we can't, there is no easy way. They're neither entirely metabolic nor entirely something else. We even have some pseudogenes, so no idea. Yeah, we haven't done, for those lines, we haven't done swaps to see which specific mutations may be responsible. Yeah, but maybe that's something we could do. Yeah. Yes.
Have you tested the effects of mutations that arose in one antibiotic environment?
Sorry, say that again?
Have you tested the effects of mutations that arose in one antibiotic environment in the other antibiotic environments? Do they have fitness costs?
Right. So, yeah, so, okay, sorry, I didn't mention that. So when we test these evolved lines in other environments, so we have another regime where the conditions are fluctuating, so it's not a constant environment, it's cancer, whatever. So in those regimes, we find that there is a fairly good things that are evolved, lines that are evolved in kanamycin do fairly well in ciprofloxacin and so on. But in these constant regimes, no, we don't see as high an increase in either MIC or growth rate across any cross test across lines. Yeah, much poorer.
Excellent. Thank you very much. I'm interested in whether you saw any reverse of you applied different methods to induce mistranslation.
Correct.
Some of them are changing tRNAs, which are much more difficult to revert, and some other touching the ribosome,
yes,
which is something that could be reversed.
Yes.
By it. So do you identify instances of reversion?
Yes. Thank you. So not direct reversion, but like I said in the strain, one of the mistranslating strains where we've made a mutation to mistranslate in a certain way, we see a secondary mutation, which we know leads to hyperaccuracy. So yeah, not in all the lines, but maybe in about 1/3 of them. we see this. Yeah. But it's regime specific also. Depends on the antibiotic.
So all your mutations, they affect the mistranslation per se, or could you have some that also affect the correction of the mistranslation?
Good question. No, so far everything we have directly affects decoding. So it either makes decoding faster, or really actually that's all it does. Faster or slower. Faster typically goes with the ribosome ratcheting before the correct Watson crick pairing has happened. So it's mistranslates. If it's slower, it's usually more accurate. Yeah. No, we don't have indirect protein quality control mechanisms mutated. Not here. We've been doing that for other things, but not. Yes, please.
Thank you so much for the talk.
Thank you.
Just curious, actually. What is the difference for you? I see that there is between alternative translation and mistranslation, because even if I see that the mistranslation is a TRV that bring a very this kind of bad recognition between codon and anticodon, and correct, this is a very explicit error concerning the genetic code. But nonetheless, there are some kind of translation that some that could be viewed as error, even in a different way, but are called alternative and not a lot of these. For example, the frame shift is when it could be an error, it's another kind of error, but sometimes frame shift or alternative cell selection are viewed as alternative translation. And so, can you, there are kind of different kind of error, but why there is this difference in
So I think this is really terminology related again, but typically in the area, so when you say mistranslation, you're right, you do include things like frame shifting, reading through stop codon. So it's not only a single amino acid substitution by another, but the ultimate readout is whether your protein sequence has changed. So that change could be at one amino acid if you have a decoding error, or it could be half your protein has a different sequence if you frame shifted or stop codon read through. Right, but what now very recently, because this is really a recent field, about 3 years ago, people did the first whole proteome measurement of mistranslation for the first time, and from that we know that decoding errors are about 75% of the total. So if you count all errors, then decoding errors they dominate.
Thank you.